Hello everybody. Today we are going to talk about skin tissue engineering. So before going into the skin tissue engineering or tissue engineered graft, we should need to know about the function of the skin. So as we all know that skin is the largest organ of the body, and mainly it does that protection from the environment, especially abrasion and fluid loss. Then we have that gives a containment for the body structure and also the organs present inside the body. It also involves in the heat regulation, like in the form of sweat, dilation, contraction and blood vessels. It also involves in the sensation of nerves, like while we touch, that conduction of the things will be passing into and will be sensed. And it also involves in the synthesis and storage of vitamin D. Further, it also acts as a blood reservoir. So it also plays an important role in excreting the unwanted substances through sweat. So before developing any skin graft or something, we need to know about that anatomy of the skin. So if we see the skin, so it has two layers. First layer is dermis and the second top layer is epidermis. So on the bottom layer is not of the part of the skin, but it also plays an role while we discuss about the skin. So that bottom layer is subcutaneous tissue, or it is called as hypodermis. So it has many parts. So attached with the hair follicles we have sebaceous gland. So sebaceous gland produces sebum. So that sebum is giving lubrication for that hair follicle, and also this is responsible for making that waterproof for the skin, and it also acts as a primary defense system. Then attached to that sebaceous gland we have arrector pili muscle. That arrector pili muscle, that is responsible for that pulling of hair. When goosebumps happens, that pulling of hair happens because of that muscle. And we have the nerve conducts. So this is responsible for the sensation, and we have blood vessels for transport of nutrients and other things from subcutaneous part to dermis, and further dermis to epidermis part. So we have epidermis. So epidermis have various layers. So if we see, the first layer is epithelial cells. So this first layer contains epithelial cells, and this is called stratum basal. Then because of the blood vessels whatever is coming from the dermis part, the blood supply is going into the epidermis, and the cells will be keep dividing. So the division of the cells goes in the top portion. Then from the basal, that goes into that next layer, that is called stratum spinosum. In stratum spinosum, the epithelial cells gets keratinized, and apart from that keratinocytes we have melanocytes, which are responsible for the color of the skin. And the next layer of that stratum spinosum is stratum granulosum. Then here the cells flattening happens, and over there we have stratum corneum, that is the final layer of that epidermis, and where that acts as the shed of that old cells. So we have in between layer epidermis and dermis is that junction, epidermal dermal junction. This is called reed ridges. So this reed ridges plays a main role in giving mechanical strength to the epidermis, and it also gives adherence to the dermis and epidermis because of this epidermal junction. Apart from that, the soluble molecules whatever is present in the dermis, the transfer of soluble molecules from dermis to epidermis is happening through this junction. So when there is a wound in normal skin, what happens is, by innate mechanism the wound will get healed. So it involves series of steps. The first step is hemostasis. In hemostasis what happens is, the blood coagulation will occur. In the blood coagulation, during that blood coagulation happen, at the wound site epinephrine will be released, and that epinephrine, once that epinephrine is released, that platelets coagulation will start, and like the complete process is called hemostasis. Then the next step next to hemostasis is inflammation. So once the blood started coagulating, the swelling and other things will happen because of the inflammation. During swelling, it also produces heat and fluid flow also will be more, and obviously the pain also will be getting. Then herein we have that defense system also will play an important role. When there is a wound, the foreign particles will try to invade into the body. So that time that leukocytes and macrophages will start developed into that particular site. So the first site of mechanism is by leukocytes. So that leukocytes will be replaced by the macrophages. So that macrophages, what it does is it will cleanse that debris of the wound. Then the next step is proliferation. So proliferation is mainly of that fibroblast, whatever is involved in that dermis. So if that wound is happening only with the epidermis part, that proliferation will happen with the help of keratinocytes. So this involves three different types or like classification. The first one is granulation. In the granulation, what happens is the cells will start dividing. If we take fibroblast or dermis as the wound site, so the fibroblast will start doubling and it will be creating multiple cells, and after that once that cells start dividing, obviously the size of the things will come down and it will contraction will happen, then epithelialization will happen. The next step is remodeling. In the remodeling, what happens is that fibroblasts will develop a extracellular matrix. That extracellular matrix contains collagen fibril, proteoglycans and other fibronectins. So fibroblast initially will produce that procollagen. That procollagen will twist together and that form a strong collagen. So many collagen molecules will come together and they form a very strong network. So because of this network only we get the fresh or regenerated tissue. So if this collagen is not twisted and it is not forming proper network, what happens will be like that skin tissue will not, that mechanical strength of the tissue whatever is formed will not be equivalent to the normal skin. So if we get a wound, we ourselves will be able to see such kind of difference. So when that tissue starts forming, that strength of the tissue will be very smooth. So once that new tissue is formed, we will be able to see that strength of the tissue will be equivalent to the earlier tissue. So this is what happening. The reason is that collagen remodeling, that is what happens at that site. So this is normal mechanism of wound healing. So when this normal mechanism is in few conditions, this normal mechanism will not happen. So the first mechanism is like poor circulation. When there is poor circulation of blood into that wound site, that normal wound healing will not happen. And when there is a edema or insufficient nutrients, that time also the normal wound healing process will not occur. Then when there is a repetitive trauma to the wound, in that time also the wound will not get healed by its own mechanism. When there is a extensive wounding, then also that wound will not heal. Then the burn lead to deep wound. If there is a deep wound, it is very difficult for that wound to heal. Then the next reason could be extensive skin loss due to infections such as necrotizing facilitases. Then we have when there is a surgery, so during surgery also we get skin loss. So that time also it is very difficult for that skin to regenerate and wound to heal. So the probable solution for these, like when there is innate mechanism is not happening, the solution could be induced primary healing. So induced primary healing will be like, consider we have wound here like this. So in induced primary healing, what we do is this portion and this portion will be combined together like this way, and they put some stitches over here, so that will be totally covered. So when this is happening, we will be able to heal. But when that wound bed size is more, it is very difficult to bring that both the sides of the wound and to bring that down into that portion. Then the second step is delayed primary healing. So, in the delayed primary healing only we will be talking about that skin graft or tissue engineered based product. So, in delayed primary healing what happens is, so consider we have a wound like this. So, this both the skin cannot be brought together and we cannot stitch it. So, for making that fibroblast to grow, we need to keep something over here. So, that will be achieved by the graft whatever we are developing. So, classification of skin involves two grafts, like first one is split thickness graft and the second one is full thickness graft. Split thickness graft involves only the epidermis regeneration. Full thickness graft involves the epidermis together with the dermis regeneration. So, nowhere that hypodermis is involved, as I told in that anatomy part, hypodermis is not part of the skin, but it plays a role when we discuss about that engineering thing. We have is skin graft, we have autograft. Autograft is from the own patient sample, they will be taking the tissue and they will be placing it. Isograft, genetically equivalent sample will be taken and from the species, same species, the graft will be taken. In the xenograft, from one species to other species, it will be transferred. So, among this autograft is advantageous because of various reasons, and the sources for split thickness graft could be like stomach or thighs or backside of the buttocks, and like as I told that split thickness graft involves only that epidermis part. In the epidermis part, what they do is they use dermatome to cut that splice. So, that splice will be further what they do is they make mesh into it and they will be placing into that wound site. Then we have sources for the full thickness graft. So, in the full thickness graft what they do is they will cut the graft from the back side of the head and buttocks and also armpits. From these portions they can get the full thickness graft. So, full thickness graft is like excision of that complete part of that skin. It is not like we do not need to use any dermatome or any other specialized instrument to cut that splice, whereas that complete skin will be taken. So, they will be excising that portion and they will be placing into that place wherever it is required. So, when they do the surgery, like after cutting the portion from the particular part, what they do is they will give some pressure onto that place and they will be placing a support like this way. So, this is called a bolster. So, they will keep the bolster and they will stitch it. So, the reason for doing such thing is, if you do not keep something over that portion wherever that graft is placed, that will not be in that position for long time. So, for making that sure, they are keeping some bolster into that surgery wherever they have done, and they will be covering it by stitching. So, advantages of this skin grafts is like it is taken from the own body. So, it is biocompatible and it is non-immunogenic. It also has disadvantages. So, the main disadvantage is like that is taken from the own body. So, obviously it will give pain to the patient. And then another thing is it will again create secondary wound. So, that secondary wound healing will be again a problem. So, these are all the drawbacks whichever we get from the autograft based skin graft. So, what could be the solution means, like we have come across from stone age to now we are in the plastic. So, in the plastic age we are using multiple polymers and we are using polymers. We can use polymers for various good applications like these kind of things. So, we can make some prosthetic implant. That prosthetic implant can be made with the polymer. So, with that polymer can be used as a skin graft. Instead of that graft is taken from own patient body, we can use the polymer as a skin graft, because ultimately we are developing the graft just to cover that wound bed. So, once we covered it and that graft is completely biocompatible, the cells will start infiltrating into it and it will try to form the normal structure of the skin. So, this is how that will work. So, consider this is our skin graft, and that skin graft is placed into that wound bed. So, once it is placed and it will be stitched, or they will be doing some dressing. Then once that dressing is completed, obviously that wound healing will occur and that will form the normal skin. So, global tissue engineering market involves highest market is based on the skin. So, once we develop that skin graft, so that can be used for two different applications. The first application could be that skin graft can be used for wound healing. As I told, the wound whichever cannot be healed by own innate mechanism, that type of things need some graft. So, for such kind of applications we can use this skin graft, and another type of applications is like for cosmetics related things. We are developing so many drug and molecules. So, for checking or screening that drug molecule, we are using so much of animals and humans. So, that involves ethical considerations and many problems. If we are able to develop a skin graft equivalent to the normal skin, so that skin graft itself can be used for drug screening applications.
Hello everyone, I am Vasudha, an MS student in Dr. Vignesh Muthuvijayan's lab. Today we are going to discuss various design strategies that have been used to synthesize tissue engineered skin grafts. Artificial skin is designed in two stages, stage 1 and 2. Stage 1 is applicable to short term acute use, while stage 2 is applicable to long term chronic use. Stage 1 deals with physical and mechanical aspects of the design, while stage 2 deals with biochemical aspects. In stage 1, the major critical graft properties that are considered include bending rigidity, surface energy which affects the wetting of the graft, moisture flux rate through the graft, tear strength and blood compatibility. The critical wound bed properties such as viable tissue are of considerable importance, and the critical properties at the graft wound interface include wetting and the peel strength, and the clinical functions that are most important at this stage are infection and fluid loss control. In the stage 2, the graft properties that are important are biodegradability and non-antigenicity of the membrane is crucial. Also porosity of the membrane, the pore size of the membrane, the thickness and the blood compatibility. So, the desired events in the graft lifetime include migration of non-inflammatory cells such as fibroblasts and keratinocytes, synthesis of neodermal tissue and also the metabolic disposal of the graft. So, in this stage, other than the infection and fluid loss control, contracture and scar control are considerably important. So, there are different physico-chemical and mechanical aspects that are important for achieving effective wound closure. So, in this representation, you can see the formation of air pockets because of ineffective wound closure. In figure B, you can see that due to excessive flexural rigidity of the graft, there is formation of air pockets between the at the wound and graft interface. In figure C, due to excessive shear stresses, there is formation of air pockets. In figure D, that is due to excessive peeling forces, and in figure E, due to excessive moisture flux rate through the graft, there is dehydration of the membrane at the corners, so which causes lifting of the membrane. In figure F, the moisture flux rate is insufficient. Due to that, there is excessive fluid accumulation which can cause edema. So, all these should be prevented to achieve effective wound closure. So, what is the long range design objective for artificial skin? You should design a membrane that provides satisfactory wound closure to prevent infection and fluid loss. The graft or the membrane should be solid, degradable and non-antigenic, and also it should provide early opportunity for cell migration and connective tissue synthesis, that is collagen deposition inside the membrane or graft. So, the lifetime of the membrane is a very important aspect. So, here Tb is the time constant of biodegradation and Tl is the time constant for normal healing of the skin incision. So, the Tb, that is the biodegradation rate, has to be optimized, because if the Tb is much lesser than Tl, then there is ineffective wound closure, that is the membrane is getting degraded before the healing process takes place. Also, if it is much higher than TL, then it has been observed that there is fibroid tissue formation under the graft. The next aspect is porosity. Porosity is crucial to allow cellular migration into the graft and also important for the exchange of gases and nutrients. Porosity is also important to allow the cells that participate in wound healing process to migrate into the graft, majorly being mesenchymal and epithelial cells. These cells have a size of around approximately 10 micrometers. So, the pore size of the membranes should be larger than this to allow cellular migration. Also, a different type of cells invade the initially non-cellular membrane. This is an image of, this is a scanning electron microscope image of a silk fibroin chitosan blend scaffold. So, you can see the interconnected pores in the image. Cell motility through the graft depends on the availability of gases and nutrients. So, the conditions under which diffusion alone suffices to maintain the supply of nutrients to the cells that are advancing through the graft is estimated using a dimensionless number S. So, in this equation, gamma is the rate of utilization of a critical nutrient by the cell. L is membrane thickness, distance along the membrane thickness along which the cell has to migrate itself. D is the diffusivity of the nutrient in the membrane. C naught is the nutrient concentration at or near the surface of the wound bed. So, LC is the maximum path length along which cells can migrate without requiring any more nutrient than supplied by diffusion. So, if your membrane thickness L is approximately equal to LC, then the cells can migrate without requiring nutrients other than that supplied by diffusion. However, if the membrane thickness is large, much larger, then what happens is you need an additional mode of nutrient supply. So, then there is need for migration of endothelial cells and vascularization, which is an important aspect in skin tissue engineering. Endothelial cells have a migration rate of around 0.4 millimeters per day and fibroblast cells around 0.2 millimeters per day, and this rate is usually insufficient to cover the surface of the graft in a period comparable for wound healing. So, an alternative approach that is used is to culture epithelial cells on the membrane surface at an appropriate time following grafting. So, the materials used for skin tissue engineering have to satisfy certain needs. So, major of them include providing a barrier layer of renewable keratinocytes and allow them to securely attach to the underlying dermis, promote vascularization and also provide elastic structural support for the skin. So, the biomaterials used for skin tissue engineering can be an epidermal cover. which can deliver cultured keratinocytes. So, this is the upper thin layer of the skin. This usually is involved in prevention of infection and fluid loss, or it can also be a replacement of the dermis, which is the underlying thicker layer. So, this layer is mostly composed of fibroblasts, or it can also be an epidermal and dermal replacement, which is usually a split skin graft. Also, the tissue engineered skin can contain appendage constructs such as hair follicles and sweat glands. The presence of these helps us mimic the natural skin more closely. Also, the pre-vascularized constructs can be provided to supply nutrients to the graft. Like we discussed, if the thickness of the graft is much higher, then diffusion alone is not sufficient to provide gases and nutrients. In the next lecture, we will look at different materials that can be used for skin tissue engineering, and we will also look at various fabrication techniques that are used to synthesize tissue engineered skin grafts.